Welcome back to the lecture series in nanotechnology application in agriculture. So, in the previous class we talked about some of the diagnostic kits, especially we talked about the micro cantilever and nano cantilever fabrication and how such small devices could be used for detecting the muscle physiology and understanding the muscle physiology and testing any kind of drug without getting into the animal system. So, this is one of the area where there is tremendous amount of work which is going on across the world, is how far we can avoid use of animal for testing different kind of drugs, different kind of molecules or series of cosmetics and all these sorts of things. So, we will come later into that, but before we proceed further, let us do a summary of the different kind of nanomaterials which have found applications. So, to start off with, so as we are all aware of the challenges what we all face because of excessive use of antibiotics, and especially in the animal world the use of antibiotics is pretty rampant. So in our country also among human, like wherever, even if it is not needed, doctors will recommend, okay, take an antibiotic. The problem is that when you take excess antibiotics, you become resistant to several set of pathogens over a period of time. So, in animal production there is no exception to this rule, and incidentally the excess antibiotics comes in the food we derive from animal, whether it is milk, whether it is meat or any kind of stuff what we are deriving from animal is very badly affected because of the antibiotics. And not only that, now it has been observed that several honeybees colonies and all are also affected because of different herbicides which are excessively used in the plant kingdom. So, the new wave which is slowly coming is called the biocides, use of biocides, and there are several nanomaterials which are found application as a biocide. So, to summarize with, so some of the nanomaterials, like if we talk about gold and copper, which are essentially your metal nanoparticle, gold and copper nanomaterial found applications as a biocides for water treatment. Okay. Water treatment. Similarly, so these are acting as biocides. Okay. Similarly, casein micelles. Those of you who are not aware about casein, casein is a milk protein, one of the very abundant milk protein. So, casein micelle, which is a natural source. So, the experiment which was done was determining the storage capacity and stabilization of encapsulated bioactive compound and their bioavailability. This is used for nutrient delivery. So, almost like, and even this could be used for drug delivery too. Then you have lipid nanoparticles, lipid NPs. So, these are used for simulated diazotization assays to test the bioavailability of loaded compounds. So, these are used for, again, for nutrient delivery. Cruciferine nanostructure, these are used for same cruciferine for nutrient delivery. These are used for encapsulation of the material. Then you have calcium carbonate and calcium citrate. Calcium carbonate and calcium citrate. So, calcium carbonate and calcium citrate metal nanomaterials are used for again for different kind of nutrient delivery applications. Similarly, you have silver. Silver is an eco-friendly biocide material, Ag. So this is a biocide in animal systems. Copper, as a metal copper, this is used both as, we have already talked about biocide activity, and this is also used for nutrient delivery. Okay. Gold is used as a biocide, we have already mentioned it. Then you have chitosan polymer, which is used as a biocide. Okay. Similarly, there is another molecule called triclosan. Triclosan has anti-malarial activity, as well as triclosan is used as biocides. Iron oxide nanomaterial, and this is not an exhaustive list. These are some of the examples I am putting forward. So iron oxide is used for multiple application. So this is used for imaging applications in animal systems. So in a way, it's a diagnostic stuff. Then you have the carbon, that is glucose and sucrose delivered nanostructured. These are used for different kind of drug delivery system. And they are at times used for as a capping agent in different kind of nanomaterial. So similarly, mesoporous silica is used for spatial imaging and drug release in the body. So these are the diagnostic part. Drug release. Similarly, poly-L-lactide and poly-D-lactide and polyacrylic acid. Poly-L-lactide, poly-D-lactide. These are used for delivery of therapeutics, again drug delivery. So in other words, these are all the molecules which are used for controlled release. You have albumin dextran. Albumin dextran is used to bind hydrophobic drugs to create aqueous solution. Bind hydrophobic or water hitting drugs in an aqueous solution. In other words, they are increasing the solubility of these kind of material. Zinc oxide has to be handled very carefully, because zinc oxide has shown in several studies it has a toxic effect on livestock sperms. Similarly, titanium oxide. So these are some of the molecules, like titanium oxide is used for water purification, but one has to be very careful because they have negative influence on the sperm quality of the animal. Similarly, antibody coated or lectin coated iron oxide for nanopurification of sperm. Antibody coated or lectin coated Fe2O3. This is used for purification of sperm purification. Similarly, you have silver metals which, as we have mentioned, it has a biocide applications, biocellulose. This is also designed as an antiseptic, collagen stimulating for wound healing. So what we observe is that this whole list is pretty exhaustive as we go through. Like in the whole world of animal production, there are two aspects which both, of course, not only animal, both in the plant also in the agricultural production system. So one, what we kind of, if I had to classify, so any kind of, say, animal production, so one part where nanoparticles or nanomaterials are getting applications is the production part. Okay. Or in terms of production, we talk about growth, metabolism, animal health, milk production, milk, meat, egg. Okay. This is one aspect. Apart from it, there is another aspect in production, which is the supporting aspects. What we are talking about is the diagnostics. We have talked about several diagnostics. There is another side of the story which is kind of an extension of the diagnostics, is novel tools. There we have talked about cantilever systems and all this to detect the muscle physiology and several other aspects. Here, among the novel tools, another area which finds tremendous application is animal cell culture for assessing the nanomaterials before conducting animal trials. This was in the very beginning of the class I was telling you. So this is another area which is getting tremendous significance for last several decades, where there is a need, there is a thrust that we may need to figure out ways by virtue of which we can minimize the application of the drug in the animal directly. Instead, if we evaluate the effect in an in vitro setup, say for example there is a reproductive drug, so if we wanted to test it, we should be able to test the drug on sperms, should be able to test the drug if it is a female, related to female reproductive system, we should be able to have those tissues cultured in a dish and we could test it. Or say for example we say there is a particular nanomaterial which will have applications, say in hormonal, changing the hormonal profile. So what we can do, we could isolate those cells which produces hormones, neuroendocrine cells or endocrine cells, and we can test the particular drug or particular nanomaterials to see whether it is really doing like that. That's not essentially it's going to happen. Once you get to the animal, the story may completely change. But even before we move to the animal, there is a need to explore the wide area of nanomaterials which are coming up into the cell culture dishes. So this is one area where there is a lot of work which is going on. So the part 2 of this story is whatever the products in terms of, say, these are the products which we are deriving from animals. Preservation. And this is one area. The other one is, of course, when you talk about animal health, sperm. This also falls under preservation, because if you get a good quality sperm, you prefer to store that sperm for artificial insemination and maintaining that particular quality for a prolonged period of time. Now, for that you really need to preserve the sperm. So, as we were talking about, there are nanomaterials like zinc oxide, titanium oxide, and why these are very critical is that these are the molecules which are, or nanomaterials which are used for water purification. Now, you realize that something which is used for water purification, because titanium oxide is a photocatalytic semiconductor material, okay, and we will talk about it once we talk about water purification, how this could do. Photocatalytic, okay, so this could split water even, okay. So, what is important is that while we are using these kind of nanomaterials, one should be careful that it is not affecting the physiology of the animal. So, the same thing may affect the sperm. Now, again, if you remember, when we talk about Ceria, I told you there are a couple of contradictory studies which says that Ceria is not that effective, but that is possible how those are being synthesized. So, synthesis. So, what I expect from you people is that you should have a very holistic view from how the material is being synthesized, how it is being applied into the animal system, because this list what I am showing you could go on and on. There are so many studies happening, but really to look at them very carefully and dug out the key information is one of the challenges, okay. So, what I expect from you guys is that look through any paper you pick up, any manuscript you pick up, look through, or any experiment you design, look through it carefully and try to rationalize it, what has happened and what has been interpreted, and where possibly things could have a very long term effect which is not really mentioned in the literature. So, if one looks at it very carefully, one will be able to figure out what all experiments could be done to justify some of the claims which are being made. So, having said this, from here we will switch gears, we will move on to the zone of preservation. Our next couple of classes will be on food preservation or these animal product preservation, and what are the basic aspects what has to be taken care. So, I will close in here. In the next class we will move from the food preservation. Thank you.